Welcome to Decoding Healthcare Research. I'm your host, Efrain Riveros, Dr. E.F. Rain. In this episode, we will talk about longevity, aging, and the important role of mTOR inhibitors. We have an expert here with us. We have the privilege of talking with Dr. Dudley Lamming. He is an associate professor of the Department of Medicine, Division of Endocrinology at the University of Wisconsin-Madison. Welcome, Dr. Dudley.
Thanks so much for having me.
Thank you so much. So we are going to start discussing different aspects related to aging. One of them is going to be central here and is the mTOR. So just to give a little bit of background, in our podcast, we have been talking about exercise and how mTOR is central when it comes to protein synthesis in the context of development of muscle hypertrophy, specifically after resistance training. But the mTOR goes beyond the role in exercise. And the mTOR is definitely involved in the different processes of aging, and this has been studied in different animal models and in humans as well. So my first question before we go straight to talk about mTOR is what is your line of research? When did you start investigating about this particular topic of mTOR in aging?
I first started investigating mTOR and aging probably around 2006 or so when I was in graduate school. At the time, we were focused on the TOR homolog in yeast, which is just called TOR. And then more seriously, I sort of devoted myself to investigating the role of mTOR in aging in about 2008 when I started my postdoctoral research. And since that time, mTOR has been sort of one of the central focuses of my research program.
Yeah, for us and for our audience, we are not experts in the topic. So let's start describing a little bit about what mTOR is and in what metabolic pathways is it relevant.
Well, I think it's probably easier to say what mTOR isn't relevant to, and mTOR seems to have a role in virtually everything. But fundamentally, it's very important for a cell or an organism to know when food is available and when conditions are good for growth. And so when you think about what's needed to duplicate a cell or to increase the size of an animal or a person, what you need is building blocks. And so you need building blocks such as carbohydrates that could include glucose, dietary protein, and specifically the amino acids that make that protein up, all different types of fats and lipids to make cell membranes and for other purposes, and the ability to make parts of a subcellular machinery. For instance, you need to make nucleotides in order to replicate DNA. You need to make new ribosomes if you're going to be making a new cell. These are the protein factories in the cell. And you need to be able to build more factories. And so it's very important for a cell to know if conditions are appropriate for growth, because if there's limited food in the environment and a cell starts to undergo the process of growing, it might not be able to complete it. There needs to be sufficient food. And in the context of an animal, inappropriate growth can also occur in different tissues, right? That's essentially what cancer is. Cancer is cells growing even though they're not supposed to, even though the body is sending out signals saying you shouldn't be growing, they can't receive those. And so mTOR serves as a central regulator of all different types of nutrient and environmental cues. And so if all of the conditions are permissive for growth, so if you have enough glucose. Glucose sensing goes down to mTOR. You have enough amino acids signal to mTOR. You have enough of different types of lipids and cholesterol, and you have growth factors that are permissive for growth as well. If you have all of these signals coming in, mTOR integrates them all and says, yes, I'm looking across the entire range of these nutrient signals, and things are good. So it's time to engage anabolic processes. So mTOR then regulates things like protein translation. It regulates building new ribosomes and synthesizing new ribosomes. It controls synthesizing new nucleotides for replicating DNA. And so virtually everything that a cell needs to grow and replicate, the activation of that requires mTOR to be on. And mTOR is going to be on when environmental conditions and the environment in terms of hormones and so on is all saying it's permissive to grow, it's okay to start growing or dividing. And so conversely, if anything is missing, if you don't have enough amino acids or you don't have glucose, then mTOR should be turned off, and that should give a signal to the cell that it's time to conserve resources. And it'll throttle back, making new ribosomes or new nucleotides. It'll upregulate processes like autophagy, which finds damaged organelles and damaged proteins and helps recycle those components. And it'll say, if there are other very energetic requiring processes in the cell, maybe we should turn those down or shut them off entirely until conditions are better. So in a nutshell, that's what mTOR is. It basically switches between growing, a state where everything is growing and active, and if things aren't okay for that to happen, it turns that all off and starts activating processes to conserve energy and recycle components.
So it must be conceived as an anabolic effector, if you will, right? So the mTOR you mentioned is activated by the presence of different nutrients and a hormonal milieu, if you will. So what kind of hormonal signals activate mTOR under physiological conditions?
A ton of different things, but the sort of most obvious and acute is insulin. So insulin is essentially a signal of the fed state for an organism. And because when you eat, you're eating a meal and that's going to contain sugars, and those sugars are going to stimulate the release of insulin. And conversely, if you are starving or fasting between meals, you're going to have very low insulin. So insulin is probably one of the most acute ones, but there's a large number of other hormones that directly and indirectly feed into mTOR signaling. Many things that signal nutrient state and things that also signal inflammatory state like TNF alpha. So I'd say, it really is easier to say what doesn't regulate mTOR, because one way or another, virtually everything in the cell and in the body seems to feed into regulating mTOR activity. That's its main role. Its main role is to be the central sensor.
Yeah, it's very central. I understand. So once the mTOR is activated, what happens after? What is the cascade of events that ensue once this is activated?
mTOR is a protein kinase, which means it phosphorylates other proteins. And then these proteins in turn either do things or directly or might activate additional protein cascades. And so some of the most common effectors of mTOR signaling that are characterized include S6K, S6 kinase, which regulates the phosphorylation of S6, which is part of the ribosome. 4-ABP1, it's a binding protein that regulates translation initiation. And ULK1, which is a component of the autophagy machinery and actively negatively regulates autophagy. But there are many, many different mTOR substrates that have been described. I can't tell you all of them off the top of my head, but it would not be surprising if it's an excess of 20 to 30 different substrates that have now been described that are regulated by mTOR.
In your paper, you mentioned that the pharmacological inhibition of mTOR is associated with longevity, and this has been demonstrated especially in animal models. But before we talk about the evidence, I have a question. So it's also true that when you do exercise, you need mTOR. mTOR is activated in the presence of amino acids to build protein. As you mentioned, the environment is adequate for the protein synthesis to happen, and mTOR is central there. So it seems to be a good thing, right? Having the mTOR activated. So at what point having a hyperactivation of mTOR becomes a problem?
Hyperactivation of mTOR is probably a problem in a lot of different contexts. Sort of one of the mTOR is sort of one of the problems, though, is not necessarily that mTOR is active. It seems to be probably that mTOR doesn't turn on and off correctly. And so one of the things that we see in aging skeletal muscle, in aging brains, in the context of Alzheimer's disease, probably in other tissues as well, is that instead of turning on and off as you go through the day and you go through cycles of feeding and fasting between meals, mTOR signaling seems to become sort of stuck constitutively on. And so it doesn't go turn on and off in an appropriate fashion. And so, that probably is one of the major issues, rather than that it is occasionally sometimes high. It's that there aren't periods where it's turned off as well. And not to say by any means that I'm an expert in mTOR in muscle, but I'll say another cue that feeds into mTOR in muscle is mechanostimulation. And so as you exercise, that also regulates mTOR in skeletal muscle. And so that's another cue that mTOR needs to be on following exercise in order to help rebuild some of the skeletal muscle that has been damaged during the process of exercise.
I see. And understanding the importance of mTOR, and there has been a lot of enthusiasm after the reports showing that in animal models, inhibition with rapamycin of mTOR leads to a longer lifespan. So what is rapamycin, and what are the mechanisms there for non-experts like myself?
So rapamycin is a small molecule, and it falls into the category of a macrolide antibiotic that was discovered a number of years ago now, originally in a soil sample that came from Easter Island, or as the natives called it, Rapa Nui, back in the 1970s. And this molecule has some really interesting properties, particularly its ability to slow growth and proliferation of a wide variety of different cell types. Strictly in terms of, presenting it simply, but for a scientific audience, rapamycin binds extremely tightly to a protein in the cell called FKBP12. There's some other FKBPs that probably rapamycin binds to as well. And this binding protein and rapamycin form a dimer that then binds very tightly to a specific domain of the mTOR protein kinase itself. And by doing so, it inhibits mTOR function very quickly and not irreversibly, but essentially irreversibly for most practical purposes. mTOR in mammals is found in two distinct complexes, mTOR complex 1 and mTOR complex 2. And one thing that seems to be important for the action of rapamycin is that in mTOR complex 1, this domain on mTOR that is bound to by rapamycin FKBP is on the surface of the complex, and so it's very accessible. Whereas in mTOR complex 2, it's buried inside the complex and isn't accessible. And so rapamycin acutely, within minutes in cell culture, probably within minutes to an hour in animals, inhibits mTOR complex 1 very strongly. mTOR complex 2, on the other hand, is not really inhibited until rapamycin has been around for a long time in an organism or on a cell. And that's due to the fact that this binding domain is buried underneath its surface.
So it's less available. And in terms of functionality, what is the difference between the two complexes in terms of like consequences after you activate them?
So the nutrient sensor I described really is a very, very sort of specific to mTOR complex 1. And most of the nutrient sensing information about environmental nutrients goes directly into mTOR complex 1. And that's some of the substrates that I described. mTOR complex 2 acts as a tuner of essentially insulin signaling. And so downstream of mTOR complex 2 actually comes mTOR complex 1. And so inhibiting mTOR complex 1 is probably where a lot of the benefits come from based on genetic experiments that I and others have done. Whereas inhibition of mTOR complex 2 actually seems to be associated with some side effects of rapamycin that might be deleterious. Primarily insulin resistance, but some other consequences as well. And so we do have these two different complexes where most of the clear effects and clearly beneficial effects seem to come from mTOR complex 1 inhibition.
And rapamycin is a non-selective inhibitor, right?
So rapamycin is a non-selective inhibitor of the two mTOR complexes, but because of the structural differences, it tends to acutely inhibit mTOR complex 1 much more quickly than any inhibition of mTOR complex 2 occurs.
So it would be fair to say that the effect of mTOR complex 2 is dose dependent. So if you fall short a little bit in the dose, you may be able to affect only the complex 1?
Well, more likely, it's related to timing. Because at least in vitro, in a test tube, mTOR2 is completely stable and resistant to rapamycin. But in the cell, we know that all proteins and all protein complexes turn over. At some rate, existing proteins are degraded, new proteins are synthesized to take their place. And so the prevailing model is that rapamycin inhibits new formation of mTOR complex 2. And so if there's some rapamycin around, as new mTOR complex 2 is synthesized and old mTOR complex 2 is degraded, an increasing proportion of mTOR complex 2 will be inhibited by rapamycin. As a functional consequence of that, yes, there probably is a sort of a critical dose but you're probably getting minor inhibition of mTOR complex 2, I would guess, at most doses.
I see. And let's jump into the evidence about the beneficial effects of rapamycin in animal models. So what does the evidence say? What do the studies say, and what animal species have been studied or used rapamycin in?
So rapamycin extends the lifespan of yeast, which is not an animal. But in terms of animals, then it also extends the lifespan of worms and flies, and most notably mice. And so many different studies have been done in mice now. I would guess probably over 30 studies by at least 10 different laboratories, including the NIA's intervention testing program, have shown that rapamycin at all sorts of doses extends the lifespan of mice. There's a lot of really interesting data suggesting that it works when initiated fairly late in the life of a mouse. It seems to also work very well when it's given over the course of the lifespan. It may even extend lifespan when rapamycin is given just at the very beginning of life. And so there's rapamycin overall seems to be very potent in mice as a positive effect on longevity.
I understand that rapamycin is FDA approved for certain conditions, probably in oncology. Has it been studied in those patients who receive it for those reasons in terms of the effect on longevity?
So rapamycin most notably was originally approved, and the rapamycin analogs everolimus and temsirolimus as well, as immunosuppressants. And they are very good immunosuppressants, and they've been used as immunosuppressants for a long time. And they do indeed suppress the immune system. Rapamycin also suppresses the immune system of mice. Now, one potential, thing to think about in terms of the translational applicability of rapamycin is that mouse studies, for the most part, are being conducted in laboratory environments where there are no or few pathogens. Whereas people in the real world who are taking rapamycin have side effects, particularly if they're taking it for immunosuppression following organ transplantation. And at least at those doses, as well as in doses used in oncology, rapamycin has a variety of different side effects, most notably an increase in the risk of infection.
So, but there hasn't been a study addressing the issue of longevity in human beings yet, right?
There is not. And, in terms of interpreting, existing data from people who have gotten this for various cancer treatments or from immunosuppression, these are people who are, generally speaking, are probably pretty sick to begin with. They may also not be taking rapamycin for that long. Rapamycin in the immunosuppressive context, people who take that are usually monitored for a variety of side effects. And if those side effects occur, they might be switched to a different immunosuppressant. Same thing with cancer. You might be using rapamycin for, a particular cancer or cancer-like disease, but, that might not be for very long, even though it might improve survival in cancer. In absolute terms, it may not, be a cure or anything like that. And if it was, you'd probably stop taking it.
Yeah, exactly. And this is probably for now a limitation that we have to translate all these findings that are very promising into the human lifespan. But let's focus now on the mechanisms underlying the beneficial effects of rapamycin. The first one that I want to discuss has to do with the effect on protein translation, and I am trying to use this segue based on the effects on the muscle in exercise, but it happens in every organ. So, what does mTOR do to the protein translation?
Well, sort of the conventional wisdom would be that it inhibits protein translation. The sort of more refined variant on that would probably be that it shifts the type of proteins that are being translated, rather than having a dramatic effect on bulk translation overall. But it certainly seems to affect the types of proteins that are being translated, and that has something to do with the specific 5' motif of the mRNA that's being translated, as well as mTOR's control of the 4E binding proteins. And so there's a shift in types of things that are being regulated, and at the risk of sort of anthropomorphizing a cell, you can sort of predict what some of those types of things are going to be. So mTOR inhibition is going to make the cell think that there's a shortage of various things, such as amino acids. So what are some of the mRNAs that are upregulated in translation when you treat with mTOR? Well, amino acid transporters that promote amino acid transport into the cell are going to be upregulated. Synthesis of non-essential amino acids will also be upregulated, and recycling processes are going to be upregulated. And so, the translation of proteins involved in autophagy and the activation of autophagy and components of the autophagy machinery are going to be upregulated. And conversely, you're going to see a decrease in the translation of things that promote anabolism.
So it is expected, I'm stopping there with the autophagy. So it is expected that mTOR normally inhibits autophagy?
mTOR normally inhibits autophagy.
And
when
that
mTOR
process
is, and when mTOR is inhibited, then autophagy is upregulated.
And what is the beneficial? So this is for our audience and for myself. So what is the health benefit of having the autophagy continue?
The sort of easiest way to describe it is in the context of something like Alzheimer's disease, where it's very clear that there's an accumulation of damaged or toxic proteins like amyloid beta and tau. And at least in theory, autophagy should result in the clearance of these proteins that are deleterious. More strictly related to aging as opposed to something like Alzheimer's, there's all sorts of proteins that acquire oxidative damage or other types of damage with time in the cell. And clearing those out and or replacing them with new functional proteins is probably going to promote all aspects of cellular function, which is probably going to, quote unquote, slow aging, if you will.
Mm-hmm. Mm-hmm. That's very interesting. Also, the immune system. So you mentioned that one of the side effects is immunosuppression, and it's actually used as an immunosuppressor in the context of transplant. So, but the effect on immunity is just a side effect, or the modulation or change in the immune function. Does it have things? Does it have something to do with longevity?
That is an excellent question, and overall, that is a question that remains to be determined. Genetically speaking, targeted inhibition of mTOR complex 1 in model organisms tends to extend lifespan, not as well as rapamycin itself does, particularly in mice, but definitely does have some positive effects. And probably in that context, you don't get very much immunosuppression, although I think it hasn't been exhaustively studied. Overall, our data would suggest, and the data of other immunology groups would suggest that inhibition of mTOR complex 2 really plays an important role in many of the immunosuppressive effects of rapamycin. And so genetic inhibition of mTOR1 or targeted inhibition of mTOR1 via future design drugs might definitely be one way to sort of tease out the benefits from the detrimental effects on the immune system.
So rapamycin is definitely works. It's very evident that that it extends the lifespan in the different models. So what do you think should happen in order to get to the point when we can only get the good effects of rapamycin and get rid of the potential side effects to make it something of practical use in humans?
I mean, overall, and I'll say this with, the conflict of interest that I consult for a company that's developing mTORC1 selective inhibitors. I think there's a lot of interest in finding ways to selectively inhibit mTOR complex 1 and tease out what the side effects are. Perhaps equally importantly, we don't really know what the side effects or the benefits of rapamycin in people are. We expect, on the basis of mouse research, that various different dosing regimens could potentially allow us to more selectively target mTOR complex 1 than just giving rapamycin every day, which is what generally happens in the immunosuppression or cancer context. We think that giving rapamycin on sort of an intermittent schedule would allow us to more selectively target mTOR complex 1. And, we think that that would be sufficient to give benefits on the basis of mouse data, but, we don't know that. And so our lab, as well as our collaborators at UW-Madison, as well as a number of other groups around the country and around the world, are starting now to conduct some of the, first real clinical trials of rapamycin in the context of aging and age-related diseases. And so, for instance, at UW-Madison, my colleague Adam Konopka is leading a trial where about 72 individuals are being enrolled in a randomized clinical trial to examine the benefits and detriments of rapamycin. And there we're targeting insulin resistance and diabetes. Other individuals are going to be looking at infections in the oral cavity, the reproductive system, particularly for women, and a group in Texas as looking at Alzheimer's disease and whether rapamycin could have benefits for people with cognitive impairment. And so a number of other groups are also, conducting these sorts of trials now. So I think this is going to give us some of the first real, clinical evidence of what are the benefits of rapamycin treatment in humans, and equally importantly, what are the side effects? Because many people anecdotally say that the side effects of rapamycin are pretty minor. But we know that at high doses, rapamycin can have very serious side effects. And so it remains to be determined, what the benefits and the risks and what that ratio is for people.
Yeah, and it definitely is going to come to fruition and show that it works for longevity, but not only that, but probably the disease is associated with longevity, right? Even if a rapamycin or a rapalog is able to influence the outcome of cancer, that would translate into longevity, I suppose, right?
Well, there's sort of multiple schools of thought. One is that the FDA has certainly seemed to be open to the idea that aging itself would be a targetable indication. But the sort of traditional pharmaceutical view has always been that these types of drugs would probably be most easily approved as a treatment for specific disorders. And certainly, from the perspective of showing efficacy, being able to show that you can preserve cognition in someone with Alzheimer's disease or reverse insulin resistance in someone with diabetes are things that could be expected, probably, to be studies that are completed much faster than showing that something extends the lifespan. And so I think there's probably room for both approaches, but there's strengths and benefits to either way.
Yeah, absolutely. So my next question is, now we have been focusing on the pharmacological approach to intervening mTOR, but what about non-pharmacological interventions, like changes in lifestyle that can indirectly or directly affect mTOR? Do they have room to be explored?
Well, my lab has focused on protein restriction and the restriction of specific amino acids. And we know that we and others have definitely shown that eating less protein, if you're a mouse, leads to lower activity of mTOR in many different tissues. And importantly, eating a low protein diet actually extends lifespan in mice. Now, there's a lot of questions about, the relevance of these types of findings to humans. With Luigi Fontana's laboratory, we had looked at metabolic outcomes in people on a protein-restricted diet. More recently, another group looked at protein restriction in the context of people with diabetes. In both cases, short-term protein restriction led to reductions in adiposity, improvements in blood glucose control, and improvements in insulin sensitivity in people. And generally speaking, we see very similar effects in mice. So, and epidemiologically, there are a number of different studies that suggest that in the human population overall, which of course is not sedentary, not athletic, not taking protein supplements for weightlifting or athletic performance, that in that context, the more protein that you eat, the greater your risk of diabetes, cancer, cardiovascular disease, and maybe even increases in mortality rate. So, there seems to be a lot of evidence that suggests that reducing dietary protein could have beneficial effects. There are counter studies, particularly in humans. There's a lot of concern that people with sarcopenia who are just becoming very frail at the end of their life, they may need more protein. But certainly it seems like dietary protein restriction might be one way to lower mTOR activity, and that might have beneficial effects. My lab has been working on looking at individual amino acids in this context, but other diet plans have also been reported to lower mTOR activity, like a ketogenic diet.
Ketogenic diet, yeah. And do these changes extend to calorie restriction, or is just the protein restriction?
The calorie restriction question is actually quite complicated. And in the laboratory, it's confounded by the fact that most laboratory animals are fed one meal a day. And so there's sort of a binging behavior where they eat for a few hours and are fasting for a prolonged time between meals. Of course, humans who are following sort of a one meal a day plan or intermittent fasting might be engaged in sort of a similar pattern. And in both of those patterns, you have really big fluctuations in mTOR activity depending on when you look. And there's certainly people that say that calorie restriction decreases mTOR activity. Our work suggests that might be tissue dependent, with some tissues not seeing a decrease in mTOR activity averaged over the day, but still very distinct patterns of when mTOR is active and when it is inactive. So I think that's a more complicated question that more work needs to be done on.
Absolutely, absolutely, and it's very interesting how you put everything together in the paper, and I think there is a lot of research coming about this topic in terms of pharmacological and non-pharmacological interventions. So, for your group in particular, what is coming next for your group in research?
I mean, some of the things that we have recently preprinted include work showing that protein restriction has benefits in a mouse model of Alzheimer's disease, where It slows the development and progression of both Alzheimer's pathology as well as improving metabolic health, increasing survival, and preserving some aspects of cognition. And so, that I think is very exciting. We also just preprinted a study where we are looking at isoleucine or protein restriction as late life interventions, starting in very relatively old animals, probably about the equivalent of a 60 to 70-year-old human. And we see definitely that there seem to be some benefits, and so at both at the level of physiology and at the molecular level as well. So we're really interested in trying to understand how, age impacts the efficacy of our types of interventions, and whether these types of interventions could be, usefully deployed in older individuals and not just animals who are more, or people who are younger, like most of our animal studies have been so far.
Yeah, and all those topics are very, very interesting and exciting, and we will be following up on your results. And I think this way we can come to a close. And I want to thank you for being here with us today. And I think the message was very clear. And I'm completely sure that our audience is going to enjoy this the same way I'm doing it right now. Thank you so much. Thank you so much for being with us today.
Thank you so much for having me. It's been a pleasure.
Thank you. And for our audience, don't forget that you can find the link to the paper in the description of the video. And don't forget to leave the feedback in the comment section. Thank you so much, and see you in the next episode of Decoding Healthcare Research.